Welcome, dear students. In our lesson today, chapter 5 also, we're going to get some information in differentiation between the experiments of the scientists and how they are get much more information and importance on the DNA. Let's go down the lesson. The structure of DNA. The last time we have got some experiments about the structure of DNA and from which type it consists of. As we said, it consists of sugar of 5 carbons, or ribose sugar without oxygen. Don't remember that, deoxyribose sugar, and phosphate group. And after that, they are linked together with what? With something called nitrogenous base. They are maybe adenine, cytosine, thymine, or guanine. Okay, and right now we're going to start in the experiment and as a kind of continuity to continue the experiment of Franklin. Okay, she supposed that DNA is made up of nucleotide, and nucleotide is which is made up of 3 parts, as we said: 5 carbon sugar, deoxyribose sugar in case of nucleotide, and in case of ribose sugar, phosphate group also. They are connected with what? With the sugar 5th carbon atom, and also one of the nitrogenous bases, which is what? Maybe adenine, thymine, guanine, or cytosine. The nitrogenous base, as we said, it may be what? Thymine connected with the cytosine, and adenine connected with guanine by a triple covalent bond. And in this technology of X-rays carried out by Franklin, she wanted to get crystals of DNA. Well, by passing through crystals of molecules which have a regular structure as a result of scattering of X-rays. They exposed or they subjected the DNA molecules to the X-ray or the diffraction of the X-ray, or they are called actually the diffraction technology. This produced what? A pattern or shapes that a kind of dots. They are by analysis of these dots, they got some information that the shape and structure of DNA. In 1952. Franklin produced such photographs for crystals of highly profiled DNA. After hundreds of experiments and try and error experiments, Franklin got purified and clear form or shape for the structure of DNA. Her results showed that DNA is twisted into a spiral or double helix with the bases perpendicular to the length of the fiber, the length of the backbone. So before the experiment of the Franklin, most of the scientists they don't have a much more imagination about the structure or the helix of DNA. And after Franklin experiments, after she already extracted or she got a purified dot or crystals of DNA, she proved that the DNA is twisted into what? A spiral or helix with the bases, that these bases are perpendicular to the length of the fiber or the length of the backbone. These pictures also prove that the evidence that the sugar phosphate, okay, which is ribose sugar or backbone, is on the outside of the helix with the bases on the inside. Okay, so we have here the main or the total structure of the DNA. The sugar phosphate, they are inside the structure or inside the double helix, while the nitrogenous bases are out with the bases on the inside. while the sugar base or backbone, they are outside. The diameter of the helix also showed that it must be composed of more than one strand of DNA. So we got the first evidence, okay, about the molecules in and molecules outside, and also the diameter by the diffraction technology by using X-rays which are carried by Franklin. She proved also that the DNA, it should be a double strand, not single strand. And this is the final structure they are discovered, or they are agreed about the DNA structure. It consists of what? Double strand, and each strand consists of nucleotides. The nucleotides consists of each nucleotide 3 parts. The ribose sugar, phosphate group, nitrogenous base, maybe adenine or cytosine, thymine or guanine. And the nitrogenous bases, as you can observe it, they are inside, and while the other parts, they are outside the ladder. After Franklin made her pictures, a race started. There is a competition. They are already came out of blue between scientists to put all the available data together and to get much more information about the constant structure or constant model for DNA structure. Each one of them wants to get the final answer about the structure and using the diffraction technology or the X-rays technology. But the two British scientists, Watson and Crick, and remember well, both of these scientists, they are very important because they have a great role in the science in changing the direction and in our imagination about the DNA structure. Watson and Crick, don't forget their names, were the first to put, they are considered as the first scientists, they are put the acceptable model for the DNA. And actually, the structure of DNA right now, as you're going to search for it and to observe, browse it on the internet, it was made by Watson and Crick, the British scientists. The model of DNA structure put by the Watson and Crick consists of 2 strands of DNA. So we have here a complementary of the information by the efforts of the scientists, okay? Starting with the evidences which material that carry the genetic information, protein, and this debate protein or DNA. And after that, we get the clear-cut evidence that already DNA is the carriers of or bearers of the genetic material. We discovered the structure of DNA, and by using the X-ray diffraction or diffraction of technology, the Watson and Crick, they put the final structure or the most acceptable model for DNA, okay? And they proved that the DNA consists of 2 strands. The 2 strands are arranged like a ladder. Okay, it looks like what? You know a ladder? Yes, it looks like a ladder. With the ladder's sides, the sides of the ladder, there is something, and inside the ladder, there are another molecules. Okay, the ladder sides, the sugar phosphate backbones of the 2 strands, and the rungs being the bases. I mean the rest, the remainders are the bases of the nitrogenous base. Okay, adenine, thymine, cytosine, and guanine. Okay, don't forget their names. The rung or the steps of the ladder, the steps, rung here means steps of the ladder, they may consist of either an adenine paired to the thymine, okay? A, it paired with T, adenine with the thymine, okay? Or guanine, which paired or linked with the cytosine, okay? As shown before, adenine, thymine by double bond, while in case of G and C or guanine and cytosine, triple bond. Okay. In each rung or each step, we have also base maybe on either strand. Okay, with the same strand. So we have here a ladder. The two sides of the ladder is the sugar and phosphate group, while the bases are composed of what? The nitrogenous bases. They are linked together. The pair of bases in each rung, in each step, is held together by hydrogen bond. So we have inside the structure of DNA the hydrogen bond. Adenine, it connects and links with the thymine by hydrogen bond or double hydrogen bond, while cytosine and guanine by triple hydrogen bond. Okay, two hydrogen bonds hold an adenine thymine pair together, and as we said, the guanine and cytosine hold together by what? Triple or three hydrogen bonds. Since each pair consists of one single and one double ring, again, each pair is what? Consists of one single and one double ring. And all the rungs, all the steps of the ladder are the same in width. Okay, and the backbone itself, the rest of the DNA, of the two DNA strands, they are what? They are always the same distance from one to another along the length of the DNA molecule. And we can also conclude and deduce that we have a kind of stability in the structure of the DNA according to what? The width and the length. Okay, the width between the two strands, it's the same, and also the length of the two strands are absolutely the same. They are equal to each other. They are actually, we have another word here, they are complementary to each other. They are what? Complementary. Each one completes the other side. Watson and Craig also, they showed that that hydrogen bonds to form properly between the base pairs in the DNA. Okay, they are suitable and proper for formation in the base pair of the DNA. The two nucleotides strands of the DNA molecule had to run in the opposite directions from the other strand, with the free 5' or free phosphate groups of the two strands at the opposite end of the molecule. So Watson and Crick, they didn't give us the acceptable model for structure of DNA, but they also gave us a lot of information on the way of connecting the two strands together and how the hydrogen bond has an important role in the twisting and also in the connecting and the stability of the whole structure to be more concurrent together. Finally, the whole ladder of DNA is twisted, okay, spiraled with 10 nucleotide pairs per turn. So in each turn, okay, in each turn of the twisting of the DNA, if you're going to count the number of nucleotide, do you remember the structure of the nucleotide? As we said, ribose sugar, which is 5-carbon sugar, phosphate group, and finally one of the 4 nitrogenous base, adenine, thymine, guanine, or cytosine. In each twist or each turn, we're going to count about 10, okay? About what? Again, 10 nucleotide in each turn. So, to form what? The spiral deducted by Franklin's X-ray photographs. And here the integration between the scientists' experiments, okay? They got a lot of information from the diffraction or the X-ray technology by carried out by Franklin, and they are put the final imaginary form or structure for the DNA, Watson and Crick. The spiral is composed of two strands, okay? They are going around each other, where the DNA molecule is referred to as a double helix. So it looks like as a double helix with the looks like as a ladder, and this ladder twist in each term or each twisting. We have about 10 base pair. After that, we need to study some information about the DNA replication, how the DNA replicate, how the DNA duplicate. Well, before a cell divides, its DNA is replicated or duplicated. Okay, so the replication, it's mean what? It's mean increasing in the amount. We have, as we studied in third prep, we got some information about how the cell they are divide. We studied the division of the cell. We said that we have two types of division, in somatic cells and in the reproductive cells. And actually, in both types of the division, the DNA, it is necessary to duplicated or replicated. Okay? Well, let's to get some information or to discuss this point. Each new cell receives or takes a complete copy of the original cells, the original genetic information, parental and maternal. Okay? Maternal comes from the female gamete. Paternal comes from the male gamete. Okay, and each cell gets the whole information. Okay, half from the maternal and half from the paternal genetic material. Watson and Crick pointed out that the double-stranded, the base paired structure of the DNA molecule, actually, it means that the genetic information can be replicated accurately. So they also give us a final answer for that. How the cell divides, probably they are divided by what? By replication DNA at the first, okay? And as you remember, in the studying of cell division, we said what? We said we have 4 phases, okay? Prophase, metaphase, anaphase, telophase. But before this phase, we have something called what? The initiation phase, okay? Or the starting phase. And this phase is kind of duplication of the DNA before starting the first one, which is what? Prophase. Before starting anaphase or metaphase, we have something which is prophase or duplication and increasing the amount of genetic material to prepare the cell for division. So, because the two strands have complementary base pairs, the nucleotide sequence of each strand automatically supplies the information needed to produce its partner. I mean, they complete each other. So, for example, if a protein of one strand, okay, runs amidst steps, it starts from 5 prime, adenine, thymine, cytosine, and ends by 3 prime. So it's the partner, or it's a complementary side. It's the other strand. It's gonna be you go into it to make something like matching together. So you have 5 prime on the other side, 3 prime. Okay, we have adenine, so it needs what? Thymine to connect with it. Adenine also thymine, while thymine adenine, and so on. Okay, but there's no mismatching between them. Get the difference? This is the meaning of they are complementary to each other. If the two strands of a DNA molecule are separated, I mean, if we go into destruct and destroy the hydrogenous base, the hydrogenal molecule, hydrogen bonds between two strands, each can be used as a model or a template to produce a complementary strand. Okay, suppose that you have a letter and you're going to broken down all rungs. You're going to break down all steps. Okay, each part in each strand, the left one and side one of them starts with 3' ends by 5'. The other one starts with 5' ends by 3'. Okay, each one of them gonna be work as a template. Okay, as a complementary part to the other one. Okay, the scientists also proved that this is the case after carrying out several experiments. Okay, we have also enzymes and DNA replications. I mean, how the DNA is replicated and what is the relation between presence or absence of the enzymes and the duplication or replication of DNA. The replication of DNA demands, it needs necessarily the integrated action of a number of enzymes and the proteins. So don't forget that the DNA, it wants to replicate, so we need some enzymes for what we have a lot of enzymes. Each one of them has its own function. One of them to separate the nucleotides from the backbone, and one of them to destroy the hydrogenous bond and the hydrogen bond and so on. Okay, let's to know it. For replication to happen or to occur, the following must happen, which is what? The double helix must be unwound. I mean, the already twisted. Okay, we need to get rid of this twisting. We need to rearrange it again. It looks like as a ladder. Okay, not in form of twisting. This is the first step for replication. So DNA, helicase enzymes. Okay, I need you to just imagine that helicase, it comes from helix. Okay, so case here, this suffix, I mean at the end of the word, this case or this suffix, it means what? It means helicase. It means to unwound. Okay, to prevent twisting of the DNA. Helicase enzymes, it moves along the double helix for what? To separate the two strands from each other. So the first point that we have to replicate the DNA, we need what? We need to separate the two double helix from each other. Okay, and it's gonna be happened by DNA helicase enzymes, which move along on the double helix to separate two strands from each other. Also, by breaking the hydrogen bonds between the pairs. Remember that we have we have dozens of hydrogen bonds between adenine and thymine, double and 2 hydrogen bonds between C and G, cytosine and guanine. We have 3 hydrogen bonds. All of these tremendous number of bonds, we need to remove it. We need to separate the two sides of the ladder. We need to separate the two strands away of each other by using what? The helicase enzyme. Okay, so by breaking the hydrogen bonds between the paired bases to expose the base, so they can form hydrogen bonds to new nucleotide partner. DNA, after that, the second enzyme that we have, the second one, which is DNA polymerase enzymes. Okay, as we said, we separate the two sides away from each other, and after that we need what? We need to break down each strand or each side into its molecules by using what? Polymerase enzymes, which build the new DNA strands, which catalyze, okay, or decompose the addition of monomers one by one. Okay, I mean starting from the 3' end of the new DNA strand and so on to reaching to 5' end. Okay, to be added to the new strand. A monomer must be paired to a base exposed on the opposite template of DNA strand, I mean the opposite side. So they are, it is necessary to be what? To be paired to the base. Okay, this is a diagram that shows us the DNA replication. We have here number 1. On the left side of the screen, we have the helicase enzyme. Okay, it's ready. It needs what? To separate the two sides. Number 1, it's unwounded the DNA and also breaking down the hydrogen bond between nitrogenous bases. And finally, we have here what? The polymerase, okay, to form the complementary side or part for each strand, okay, starting from the 3' till ending the other side, which is 5'. It is known that the deoxyribose or DNA polymerase can work only in one direction, from 5' toward the 3' and end. On the new strand, it is synthesizing, but actually we have said before that the two strands of the DNA or the double helix, they aren't parallel to each other. Okay, so there is a kind of twisting between each other, and one runs, I mean, in the 5' to the 3' direction, where the partner runs in the opposite direction, 3' to 5'. So there is no, we don't have the symmetrical as you imagine it, but they are what? They are opposite to each other. Okay, the side which starts with the 5', okay, it faces on the other side 3', okay, and also at the bottom, starts with the 3' and it faces what? The 3' and so on. So we have the helicase moves along for separating the two strands. It works towards the 3' end of one strand and the 5' end of the other strand. For the 3' to 5' template or the other side strand, there is no difficulty that the polymerase follows. Okay, the helicase adding a new nucleotide to the 3' end. However, this wouldn't work on the opposite strand, because we have here a very important question, that DNA polymerase cannot work from 3' end toward the 5' end of the strand, which is synthesizing. Okay, so remember that the polymerase always it doesn't work from 3' to 5' strand. Okay, the strand has to be made in a short piece or short pieces in the direction of 5' to 3', and these short pieces are soon joined together by another enzyme which is called DNA ligase. So right now we get some enzymes, which is helicase enzyme to separate, polymerase enzyme, okay, and remember in which direction, which side it can work and which side doesn't work. And finally, we need what? The small piece is joined together by another enzyme which is called DNA ligase. And this is what? This is the duplication, okay, of DNA. That we have here what? The polymerase enzyme, okay, which works from what? The 3' to 5' end, and on the same direction, of the same parallel to another enzyme which works what? One of them is to separate the other strand, and of the same side the other enzyme or polymerase, it brings the monomers or nucleotides to make the complementary side of the strand.